Children, now comes very interesting topic again related to your cell envelope only, that is ultrastructure of cell wall. And this cell wall is not the envelope of all the cells, student, but it is only the envelope of your plant cell, isn't it? You see, these are the two plant cells. Okay, student, these are the two plant cells. Adjacent, they are located adjacent to each other. And you see, student, cell wall is made up of three layers. This middle layer or middle level, this dark green layer. Okay. Second one is primary wall, this line on either side, this orange line, this is primary wall, student, and this green line on either side is the secondary wall. So these three layers, middle layer, primary and secondary layer, this forms cell wall. Tertiary wall, student, tertiary wall is rare. It is found in only some plant cells, not in all. Okay, student. Now when we see this cell wall composition in detail, student, electronic microscopic studies reveal that the cell wall is composed of cellulose, hemicellulose and pectin substances. Now when we see the electronic details, okay, so what we observe, student, that cellulose molecules are long chains. These cellulose molecules are long chains cross-linked by hydrogen bonds, to form strong bundles of microfibrils. Each microfibril in turn is consist of aggregates known as micelles. Okay, student, and which forms the framework of cell wall. This, I repeat, this forms the framework of the cell wall, isn't it, student? Okay, I repeat again, the electronic microscopic detail reveals that cell wall is composed of cellulose, hemicellulose and pectic substances. And students, you see these cellulose molecules, these red lines, are long chains cross-linked by hydrogen bonds to form strong bundles of, these are the strong bundles of microfibril, this, isn't it? And each microfibril in turn aggregates, consist of small aggregates known as micelles, and which form the framework of cell wall, isn't it? Okay. So here one more important question is lying here. This is asked in your NEET examination, that is your plasmodesmata. Okay, student. So first we have to study primary pit fields. This much portion is remaining. From this we get one question in NEET examination. I remember in 2013 or 15 in NEET examination plasmodesmata was asked. Children, so what are plasmodesmata? These are very thin protoplasmic connections. Between the adjoining cells through primary pit fields. Okay. And what is the function of plasmodesmata? Plasmodesmata facilitate movement of substance between adjacent plant cells. Okay. And what are the primary pit field, children? Primary, here the primary wall appears as a, this primary wall appears as the bead like structure due to presence of depressions, and these depressions are called primary pit fields, isn't it? And you know, the student, you know the function of cell wall, because it is non-living, so it is fully permeable. It is freely permeable. It acts as freely permeable envelope, isn't it? It is not selectively permeable. So student, this is all about ultrastructure of cell wall. That is the another envelope of your cell. Okay student, now we are studying nucleus, structure of the nucleus. Student, it was discovered by Sir Robert Brown, isn't it? And this is the structure. In the center of the nucleus here, in the middle portion, we see nucleoplasm, isn't it? These are nucleoplasm. And in this we found nucleolus, okay? Nucleolus. And in nucleoplasm, the most important structure is this chromatin network, genetic material, isn't it? Okay? And these are nuclear pore. The most important part which is to be studied in nucleus is, student, about this chromatin network. Student, chromatin network is composed of DNA bound to histone protein. That forms nucleosome. You have studied in last lecture somewhere that DNA along with histone octamer forms nucleosome, isn't it? Now student, when we see chromatin network in detail, it consists of two regions: heterochromatin, this darkly stained region, heterochromatin, and this lightly stained region, this one is called your euchromatin, isn't it? This is euchromatin. How we define, student, how we differentiate euchromatin and heterochromatin? Student, euchromatin is lightly stained region of chromatin network which is genetically active. This one, which is genetically active. It is considered to carry hereditary character. Genetically active means what, children? It is considered to carry hereditary character from one generation to another. And student, heterochromatin, just opposite and parallel, it is genetically inactive. This stained one, these are heterochromatin, children. These are genetically inactive or inert region, means they cannot carry hereditary information from one generation to another. Okay, but student, heterochromatin remain closely associated with the nucleolus. It is said that heterochromatin is remain closely associated with the nucleolus. Now student, what is the function of nucleus? First important function you have studied in your basic class. You are studying this from your basic class. This is the brain of the cell. It controls all the metabolic activities of the cell by controlling synthesis of the enzyme, isn't it? That is why it is called brain of the cell. Like our brain controls entire body, similarly it controls all the metabolic activities of the cell by controlling the synthesis of the enzyme. Okay? Then chromosome present in nucleus, this one, all this chromatin material, carry hereditary information from one generation to another. Okay? And this one, nucleolus in the nucleus participate in the formation of ribosomes and protein during protein synthesis. Okay? Protein synthesis is impossible in the absence of ribosome. Okay? So ultimately nucleolus play important role in protein synthesis also, because it is responsible for production of ribosomes. Okay students, so this is all about your what we call nucleolus, ultrastructure of nucleolus discovered by Sir Robert Brown. In this, most important points are euchromatin and heterochromatin. This is genetically active and this is genetically inactive. Okay student. Now the next topic in this chapter is structure of eukaryotic chromosome. Structure of eukaryotic chromosome, student. ठीक है, eukaryotic chromosome but not prokaryotic. Okay, so according to your syllabus, in this ultra structure of eukaryotic chromosome, student, we have to emphasize more on chromomere, kinetochore, satellite. So one by one I will explain you in detail. ठीक है student, this is the primary constriction. Constriction means, you know student, furrow like in this primary depression of the chromosome body. Here kinetochore is there. Okay. Now what are kinetochore? These kinetochore are in the primary depression or constriction. Student, kinetochore is a proteinaceous disc attached to the centromere. ये centromere है. These two dots are kinetochore and this is centromere. ठीक है. This is a center of assembly for microtubules or spindle fibers during cell division. You know very well that chromosome forms attachment with the spindle fiber during cell division. ठीक है. This chromosome forms attachment with the spindle fiber during cell division. So kinetochore along with the centromere is responsible. ठीक है. And we can observe this attachment at metaphase. ठीक है. So what is kinetochore? It is a proteinaceous disc attached to the centromere. Here this is centromere and these dots are proteinaceous disc, and this is a center of assembly of microtubules, means center of assembly of chromosome with spindle fiber. Either you say microtubule or spindle fibers during cell division at metaphase, isn't it? Means it forms attachment with the spindle fiber during cell division. Now student, what is chromomere? Student, chromomeres are serially arranged bits of eukaryotic chromosomes. ठीक है. It is serially arranged bit-like structure of eukaryotic chromosome. This green bit-like structure. ठीक है student. And how it is formed? It is formed by coiling of DNA threads. It is formed by coiling of DNA threads. Again I repeat, what is chromomere? Are serially arranged bits of eukaryotic chromosomes. ठीक है. It is formed from coiling of DNA threads. And now satellite. Student, satellite, a small chromosomal segment formed due to secondary constriction. This one. Student, this is first secondary constriction and this is second secondary constriction, but satellite is formed due to this first secondary constriction. ठीक है. A small satellite, a small chromosomal segment separated from main body. ठीक है. Main body of chromosome by what? By secondary constriction. This was primary constriction and here secondary constriction. Now what is the role of this? Student, this satellite act as chromosomal marker. Means when we observe in the nucleus, so how you isolate, how you recognize this structure is chromosome. So this satellite act as chromosomal marker and play important role in the formation of nucleolus. It play important role in the formation of nucleolus, and that total organization is called nuclear organization. ठीक है. Okay. So function of satellite, it act as chromosomal marker and play important role in formation of nucleolus. And so this complete organization is called nucleolar organization, and where it is found at short arm of chromosome and it forms nucleolus. Okay student. So this is all about your ultrastructure of chromosome, and in the next topic we will study types of chromosome on the basis of location of the centromere, and then there we will study the function of chromosome, that is very popular function, that is it carry hereditary character from one generation to another. So this is very important structure student. Remember the new words for you in this class: chromomere, kinetochore, satellite. ठीक है. Thank you.